Okay, so today we're going to move on to protein function, and we're going to talk about what proteins do. In particular, we've talked about protein function kind of from the point of the fibrous proteins which make up the scaffolding proteins, like when we were talking about keratin and collagen. But now we're going to talk about some more globular proteins, which are more responsible for binding to things. And we're going to talk really, this is really just a vehicle to talk about protein binding. And we're in particular, if I can find my mouse pointer. Oh, not sure why this isn't working. Do you have a pen? But there we go, my pen's writing. So in particular, we're going to use this kind of as a case study to talk about hemoglobin and how hemoglobin carries oxygen from the lungs around in the body and then transports CO2 back to the lungs again. And really, this is to do with, like I say, about protein binding. And one of the important things is that it's not just good for hemoglobin to bind things, it also needs to let go. It doesn't just need to bind, also needs to let go. And so it's going to have a variable amount of affinity. Its affinity for oxygen is going to go up and go down, depending on the condition. And it's the chemistry. The chemistry changes the protein, which changes the physiological thing that we observe. So first of all, this lecture, we're not really going to go into hemoglobin in this lecture. This lecture is mainly just going to be defining a few terms, first of all, to set the scene for everything. So really, we're going to be, like I say, understanding how proteins bind to other molecules, including other proteins. So for instance, when you, with your muscles, there is the actin skeleton and the myosin interact. Those are two proteins that cause your muscles to flex. But it's also things like hemoglobin interacting with oxygen or an enzyme interacting with its substrate. Now, because these are two separate molecules coming together, we can measure how fast they come together. And there are kinetic constraints. It's also something which is in equilibrium, where we have protein plus ligand comes together to form a protein ligand complex. And since it's got something in equilibrium, you've got the rate in which it comes together, which is given by a lowercase k, but it's gonna have an uppercase K, which is going to, what does the uppercase K signify in an equilibrium?
Constant.
Sorry?
Is it constant?
Constant, yes, constant, the equilibrium constant. And that is going to define where on the equilibrium it sits. Do you have more of it being separate or more of it being together as the complex? And we can, and different proteins have a different constant. Now, this is dictated largely by the shape of the protein, and the shape of a protein can change. And this is why we were doing all that stuff to do with amino acid ionization. If an amino acid is in a more acidic or basic or neutral condition, that's going to change the ionization state of it. And that may influence how it may interact with its partners. So if you have a carboxylic acid, which is protonated or deprotonated, it's going to be in two different charge states, and that will influence whether or not it's going to bind and form a salt bridge, a static interaction with a neighboring basic amino acid, for instance, a positively charged amino acid. So the positive and negative charges of those amino acids, they can change depending on the pH around it. And so that's going to change how the protein folds and comes together. And that's what happens with hemoglobin that we'll go on to next week. And we're going to apply this to the principles of binding of CO2 and oxygen to hemoglobin, but also this myoglobin. And hemoglobin is transport around the blood. Myoglobin is holding on to oxygen in the myo, referring to muscles. And it's actually more of a holding, whereas heme is holding and delivering. So we'll get onto that later. We also, okay. So when we're talking about these globular proteins, we can classify them into two different things. We can classify globular proteins into enzymes and other. And the key thing really that causes that is that if you have a substrate plus an enzyme, they come together to form an enzyme substrate complex, but then they're going to react to form an enzyme product because the substrate is going to change into product. So with enzymes, the thing that binds changes. The enzyme changes it. Whereas other proteins, like receptors, like antibodies, like carriers, such as heme, they bind, but they do not change. So neither the chemical configuration nor composition of the bound molecule is changed. They don't change. Whereas enzymes that catalyze a reaction, so it changes. So that's the difference. Enzymes is what we're going to cover later. And I believe that next week is our last week before spring break. So we'll go into enzymes after spring break. But until then, we're going to talk about the other proteins. And in particular, we're just going to talk about heme. And I'm going to mention a side thing about antibodies. And there's homework on receptors. But it all belongs to the same thing. A ligand binds, and the ligand is not changed. Unlike an enzyme where a substrate binds and a substrate is changed to a product. Whereas this is just a ligand and it stays as a ligand. Now there's a few different things that you can, reasons for this. With the, of course, I'll start at the bottom actually. So with the enzymes, of course, they act as biological catalysts, which turn a substrate into a product. And an example of which would be the enzymes chymotrypsin and lysozyme, both of which are digestive enzymes for break down your food. So that's the reason why you may want proteins to do that. But often you want proteins just to stick to something and bind to something, such as the myoglobin, which is transporting oxygen, the molecule of oxygen, ferritin, which is transporting iron, transport of ions and other molecules. So you've got the storage rather, then you've got transport. So sometimes you want to hold on to it, sometimes you want to hold on to it and put it in some other place, such as hemoglobin now, which you're going to transport, unlike myoglobin, which just stores, or a serotonin transporter, which will transport serotonin across a plasma membrane to, for example, in neurotransmission, to take it out of the synapse and back into the neuron. I've written neutron there by accident, neuron or astrocyte. It's going to transport it in, so across a membrane. So those are transporters either around in liquid or through a membrane. Defensive against pathogens is referring to things like cytokines and antibodies, which bind. Antibodies in particular will bind to the antigen. Antibodies will bind to the bacteria or the virus or something like that to deactivate or recruit other immune cells. But antibodies are proteins, and this antibody needs to recognize the site which it's of its target and bind to it. And of course, because it's going to bind to it, it's that same thing. We can have an equilibrium constant where we could have maybe antibody plus pathogen, forming an antibody pathogen complex, because this is now kind of a complex. Muscle contraction in exactly the same way. Actin and myosin walk along in order to make your muscles contract, and that is caused by a binding and a release, and a binding and a release of actin and myosin upon the actin to cause that walking of the actin along the walking of the myosin along the actin skeleton. So there's a lot of binding and releasing that's really important in how proteins work in biology. And because we're doing biochemistry, we're going to look at the chemistry which influence chemistry, how we measure this, and the chemistry which influences how proteins bind to things, because they're so critical to biology. And just to break it down simply, we start that with a protein, and it'll bind to its ligand to form a protein ligand complex. Now, a ligand is just anything which binds to a protein. It could be a small molecule, such as serotonin or oxygen, or it could be another protein. But a ligand is just something that binds to a protein. Now, a ligand is different from a substrate because a substrate is going to change into a product. A ligand is something that binds, but can and to form a complex, but it could also release to form its ligand again. So oxygen binds the hemoglobin and then is released as oxygen. It doesn't turn into something else. So it is a ligand. That's the difference between a ligand versus a substrate. Now, we've got a couple of slides, just 2 slides on just how things bind in terms of recognition. So as with a lot of things in science, earlier in your scholastic career, you'll learn about how something works. And then the next year, they'll tell you, actually, that's not how it works. That's how we thought it works 100 years ago, but it's actually different. Or actually, that's just an oversimplified version of how it works that actually kind of isn't true. Like when you do all your stuff with electrons, and then they teach you about electrons and orbitals, and then later on in PChem, they actually teach you that it's not real. And things like, or like the 2888 rule. And then it turns out that that's not real, and it's actually orbitals. And then later it turns out that that's not real. And they just keep lying to you. Well, in the same thing in biochemistry, they teach you about lock and key, first of all. And they teach you with lock and key that proteins very specifically bind to their ligands. And the ligand will fit like a lock in a key exactly into the place where it is supposed to go. And they are exactly complementary. And so they're going to be complementary in things such as size. So it can't be too big or too small. Shape, it's got to be the correct orientation, like here. It's got to be the shape that fits. Charge is important, right? Because if you've got a protein that's got a lot of negative sites, it's going to want to bind something positively charged. It's going to repel something negatively charged. Or it may be that you've got positive charge on one side and a negative charge on the other. And so you'll need the reciprocal ligand to also be arranged as such to have the opposing force on one side and one force on one side and one force on the other. And they've got to be able to line up. And the same with hydrophobicity and hydrophilicity. If you've got a hydrophobic pocket, something hydrophobic is going to bind. Hydrophilic, something hydrophilic. Something that's got regions of both is going to match to those same regions on the ligand. And that's mostly true. It's mostly true, but there's more to it that we'll go on to. But the lock and key is that the protein and the ligand will suit each other nicely to bind. And they'll form a perfect formation. What we now know instead is the induced fit model. And the induced fit model is that things will be somewhat complementary, but they become more complementary upon binding. Induced fit is that as when the protein and the ligand come together, they will both change. That's important. Where is it? Both. They will both change their conformation to better fit each other. That was really important. I gave a lecture earlier when I was talking about the TCA cycle and an example of the induced fit model, where the first enzyme of the TCA cycle is citrate synthase. And in citrate synthase, you have the acetyl-CoA and the oxaloacetate, and they come together in a condensation to form citrate by the enzyme CS, citrate synthase. And the thing is, you've got 2 substrates. You've got substrate 1 and substrate 2 come together, and you get just one product. Now, the important thing is that because you have 2 different substrates, you have to control which order they bind within the binding pocket. It needs to be substrate 1, then substrate 2. If you have it as this curve I'm drawing here is the substrate pocket. If you have substrate 2 and then substrate 1, the reaction won't work because the acetyl-CoA and the oxaloacetate, they're not in the correct orientation to react with each other. So it needs to be actually oxaloacetate first and then the acetyl-CoA. It can't be acetyl-CoA and then oxaloacetate because they won't be the right way around in order to react together. And so the induced fit model comes in well there, because the way that citrate synthase starts out, acetyl-CoA won't bind to it because the pocket isn't the right shape. The pocket is not the right shape for acetyl-CoA. So there's a bunch of acetyl-CoA and it comes in and goes back out again. It doesn't do anything. But oxaloacetate, when it comes and binds with the citrate synthase, it will change the shape of the citrate synthase to make it so it's now complementary, so that acetyl-CoA can now bind. And in that way, you control the order of binding. because acetyl-CoA can't bind fast because the enzyme isn't the right shape. The enzyme only becomes the right shape once the oxaloacetate binds fast. So that's an example of the induced fit. But in general, the induced fit is that both the ligand and the protein can, they don't have to, but can change their conformations to better fit each other and maybe future things. So it allows for tighter binding and high affinity for different ligands. So citric synthase has a low affinity for acetyl-CoA until oxaloacetate binds, and then it has a high affinity for acetyl-CoA. So that's important. Induced fit changes both of them to influence the way that things bind. So we're going to move away from that a little bit, as this is going to be a little bit of a patchwork of a bunch of things stuck together as we build up the definitions so we can go into the case study of hemoglobin next week. So next, just to describe the terms of the equilibriums. And that is, again, remember that protein plus ligand come together to form a protein-ligand complex. And they can come together and they can break apart. It's a reversible reaction. It's an equilibrium. It's a reversible reaction. And as with anything that happens, it has a rate at which it happens. Now, we've got to make sure that we don't mix up rate with constant. They're two different things. Now, if we're talking about getting in your car and driving on the 40 between Greensboro and Winston-Salem, you're going to, let's say that your friend calls you up and you say, where are you? You could say, oh, I'm 65 miles per hour. Well, that means nothing. But it's giving some information about how you're traveling. It's saying how fast you're traveling, but it's not really giving any positional information. But it is communicating that kind of how fast you are going. And from that, you can kind of work out, well, where you are. You know, I'm going 65 miles per hour. Oh, well, you've been gone for half an hour, so you'd be here any minute now. You know, that kind of thing. Whereas on the other hand, constant is the exact position where it is at that one point in time. That's if your friend calls you up and say, where are you? Oh, I'm at exit 221. Okay, that's communicating the exact position of where you are. Now, in an equilibrium, the rate is saying, well, how fast is the protein and ligand coming together to form the protein-ligand complex? How fast does that occur? Or how fast do they break apart? And that is given by the lowercase k. A lowercase k, I'm trying to draw a lowercase with my little tablet. A lowercase k equals rate, how fast. An uppercase k equals constant, where. And like I say, you have a rate of how fast they come together and how fast they break apart. Whereas the constant is, well, where are they actually sitting? What is the ratio? What is the ratio of these together? What is the ratio of protein ligand to protein ligand complex? That's that's what the constant is describing. Now, for 99% of the work, we're just going to talk about the constant. I'm just going to very briefly describe that the rate is there as well. And the rate is, you've got a rate of the reaction going forwards and a rate of the reaction going backwards. And at equilibrium, the rate of forwards is going to be equal to the rate of backwards at equilibrium. And that's kind of described by this little box down here. I'm going to clear the screen so you guys can see a little better. This equation here is just describing the idea that the rate of them coming together and the rate of them breaking apart, when things are at equilibrium, they are going to be equal. But if things are not in equilibrium, then the rates are going to change in order to get things into equilibrium. So these rates at equilibrium will be the same. But if the concentrations of the reactants or products, if the ratios of them change to throw it out of equilibrium, then the association or dissociation rate will change in order to get them back into equilibrium. That's a quite minor point of my course, but I do like to be sure to mention it, that because I want to differentiate between rate and constant, and really get together the idea that reactants and products will be in equilibrium, and if you push one of these out of order by taking some away or adding some, then the equilibrium is going to seek to get back into equilibrium by that LCP, Le Chatelier's principle. And so if you mess with one of these, the rate will change to push it into that direction. That's what's going on there. Like I say, it's a rather minor part, but I do like to communicate that. Something that I forgot to mention, skipped over just on this slide, is that when the protein and the ligand come together, the forces between them are usually non-covalent forces. There may be some transient covalent forces during a rearrangement, but generally they are, and for the majority of the time, what's holding them in place is non-covalent forces, such as the hydrophobic effect, hydrogen bond, and van der Waals forces. So these things coming together, they're held together by weak intermolecular forces, weak intermolecular non-covalent forces. And that's important because it allows it to be transient, because they need to come together and break apart. And they can't break apart again if the reaction is it's much harder for them to break apart if the link between them is a covalent link. So instead it's a non-covalent, they are not all one molecule, they're just linked by a weak intermolecular force. So I'm going to move on to the next slide. So like I said, if you have this, it's the same thing as just reactants in equilibrium with products, exactly like we did with the acids, is that you have the product and the ligand coming together to form a product-ligand complex, and they will work to get into equilibrium, whatever that equilibrium may be. If you interrupt that equilibrium, like for instance, let's just do as a little kind of quiz. So in this reaction, let's say that I decrease the level of ligand. What is that going to do to the equilibrium? Is it going to push it to the left or push it to the right? How about I ask it to the class by show of hands? How about a show of hands to say that if I remove ligand from this reaction, decrease the concentration of ligand, put your hand up if you think it moves to the left. Got a few, got 4, 5. If you can't put your hand up and you just want to shout out, you go ahead and shout out. But
to the left.
To the left. Anyone want to say to the right? You put your hands down. Anyone want to say it goes to the right? Nope. You got it, guys. If you decrease the ligand, then you're decreasing the stuff that's on this side. And so the protein ligand complex is going to break apart to replace the ligand that you've lost. It's more to it than that, but in a nutshell, that's a bit simplified, but in a nutshell, if you remove ligand, then to replace the ligand that has been removed, the protein ligand complex is going to break apart to compensate for that, and vice versa. If I've got the protein ligand complex and I increase the concentration of ligand, then that is going to I've got too much ligand, and to kind of get rid of some ligand, I'm going to react it with protein to form more protein ligand complex. So if I increase ligand, I'll push it to the right. If I decrease ligand, I'll push it to the left. So good work. So in particular, when it comes to this protein binding stuff, we talk about it a lot with association constant, and that is just the KEQ of the reaction. That's really just the normal KEQ of products over reactants, like I had before. Products over reactants which is, in this case, PL over PL. And that's just the KEQ is just the ratio between them. Now, they call it Ka for association constant. It's not the same Ka as acid constant. What I do, these are all just constant. They're all just K for constant. And then they just put a little letter after it just to help you know which constant you're talking about in that particular time. There could be Ka for acid constant, Ka for association constant, Kd for dissociation constant, all sorts of things like that. But the little letter is just to let you guys know what we're talking about in that moment. And it just so happens they used A twice. So Ka in this case is the association constant. And broadly, it ends up being a measurement of the affinity which the protein has for its ligand, whereby if KEQ, if Ka, which is a KEQ, you may have previously known it as KEQ. They're the same thing. They just renamed it. Not laser pointer. Pen. There we go. Ka. Now, if you have more product than reactant, then Ka is going to be high. If you have more PL than you have P plus L, then Ka is going to be high because it's a big number divided by a little number. And thus, that's telling you that the protein likes its ligand. If, on the other hand, it's the other way around, and if Ka is instead low, then what that's telling you in this equation is if Ka is low, then that must mean that P is low and reactant is high. So there's not much complex, and there's a lot of the ligand and protein unbound. So that's saying that they don't really like each other very much. So the higher the Ka, the more they like each other. The lower the Ka, the more they dislike each other. A higher Ka is suggesting a higher affinity and suggesting that things are, or indicating rather, that the equilibrium is further over to the right. Yeah, I think we'll just leave that there. Now, hold on to Ka for a moment, because the Ka and Kd, we often flick between the usage of them. And we're going to, we actually talk about Kd more. And the Kd is just going to be 1 over Kd equals Ka, and 1 over Ka equals Kd. And when you do 1 over a number, it just kind of means the opposite of. So the opposite of association is dissociation, and the opposite of dissociation is association. So 1 over Ka is Kd. So whatever the Kd value is, 1 over that number will be Kd. And whatever the Kd value is, 1 over that number will be the Ka. We're going to move on to that in a little bit, but just hold on to that in the background for a moment. Actually, I tell a lie. I recently reordered these slides, and I'm talking about the dissociation constant now. I think I did it. I think I did it in a different order last year, and then it was kind of confusing. So I introduced dissociation constant now. So I've already said, that 1 over Ka is going to equal Kd because it just means opposite in math. Effectively, it doesn't, but it does in this way. And remember that Ka was product over reactants, which is PL over P plus L, and it's the other way around for Kd. P plus L over PL, it's not plus, rather, it's multiplied. Is going to equal Kd. So it's just the other way around. You just flip it the other way around, because that makes sense. They're just the opposites of each other. And it also makes sense that Kd, if you've got a lot of PL and not very much if you've got a lot of P and L and not very much PL complex, then that's going to indicate that Kd is going to be bigger. If, on the other hand, you have a lot of the complex and not very much of the PL separately, then Kd is going to be smaller. So they are, it makes sense mathematically too. They're related to each other. Now, keeping in case, so keeping in mind that we're using an uppercase K for constant, capital, and a lowercase k for rate, which is just how fast it moves. And the KD value, the constant, is going to be equal to the rate of it going backwards divided by the rate of it going forwards, and the Ka is going to be equal to the rate of it going forwards divided by the rate of it going backwards. But this rate stuff is not super important for my course. It is very important, but not for my course and my exam. But this main bit here that I've drawn in the red box, that's the important part. That the association and dissociation constants are effectively ratios of products and reactants, ratios of product and ligand, protein and ligand, and protein ligand complex. They're ratios of each other that are reflecting where the equilibrium sits, which is going to tell you how much that protein likes its ligand. If it likes its ligand a lot, it's going to have a high KD and a high Ka and a low KD. So if we put this all together and apply this now to biology, in a biological system, you're going to have a lot more ligand than you are protein. And that makes sense. I mean, think of how many molecules of oxygen you have in your body and think of how much hemoglobin you have. You have a lot more oxygen in your body than you do hemoglobin molecules. It's just that there is always more of the protein than there is of the ligand that it binds to often. I mean, especially for the carriers and the things that we're talking about, there's going to be a lot more ligand than the protein. And so actually it'd be quite good to know that at different concentrations of ligand, how much of the protein is occupied, right? If I've got, let's say, 1,000 hemoglobins, which I'll give the abbreviation HB, what percentage of them are occupied in a hemoglobin oxygen complex? What percentage of them are in a hemoglobin oxygen complex at different concentrations of oxygen? And think about it for a moment. If hemoglobin didn't like oxygen very much, then you'd have to supply it with a lot of oxygen in order to encourage it to move into the complex. If we have the oxygen and hemoglobin, and they will form the hemoglobin oxygen complex, if the equilibrium sits mostly on the left, then that's saying that you're going to have to increase the oxygen by more in order to shift it to the right. So, depending on how much the protein likes its ligand is going to influence how much ligand you need in order to fill it up. If we have a certain amount of like let's say again, why is this important? Let's say you go into hospital, you get that thing put on your finger that measures your O2 sats. What that's measuring is measuring the percentage of hemoglobin that's saturated with oxygen. That's measuring how much your hemoglobin has got its oxygen bound to it. And if your hemoglobin doesn't have much oxygen bound to it, that's probably because you don't have much oxygen in your body. In order to fill up your hemoglobin, you're going to have to provide it with oxygen. And the more your hemoglobin likes the oxygen, the more it's going to grab onto it quickly and grab onto it and hold onto it. If it doesn't like the oxygen very much, then the reaction's sitting more on the left. And so more oxygen is going to be required to occupy it and shift it to the right. If it really likes oxygen a lot, then the reaction shift is sitting mostly on the right. And every time you bring in a little bit of oxygen, it's going to immediately react and move to the right. So not very much oxygen is going to be required to move it to the right. So the affinity, you can, once you know the affinity that your protein has for its ligand, you can know how occupied it's going to be with its ligand at different concentrations of ligand. And this is given by this equation here. And they use Y but often I see textbooks use theta instead. And it's just y because it's going to be on the y-axis when we plot it on a graph in a minute. But y is really means the ratio or percentage of occupied protein. The amount of occupied protein is given by the amount of protein that's occupied divided by the total amount of protein present. And so that's simply saying the amount of occupation of a classroom, like how the percentage occupied that a classroom is going to be equal to the number of people sat in seats divided by the number of people sat in seats plus the number of empty seats. So the percentage is going to be how much out of total. So, and the total can be figured out as, well, how many people have we got in seats plus how many empty seats, that's the total. How many occupied seats plus how many empty seats is total seats. And then occupied divided by total is going to be percentage occupied. That's just what it is. And that's what Y is. Y is the binding sites occupied divided by occupied binding sites plus unoccupied binding sites. Simple. Now, if we can rearrange this equation, and if you really want, the textbook tells you how you rearrange this equation, but we can rearrange this equation into this equation. I'm not going to go into why, because it's just a bunch of algebra that's not important, but effectively this rearranges into y equals L over L plus KD. And that's saying that the occupancy, that will tell you the occupancy of the protein at different concentrations of L. If you have y equals, let's just say x divided by x plus y, as you increase x, then it's going to increase y. That's all we're really saying. And it's just reflecting that at the different concentrations of ligand, how occupied is the protein going to be? And it's going to be the amount, the ligand supplied divided by the ligand supplied plus the rate of dissociation. It's going to describe the ratio of occupied protein at a given concentration of ligand. But you need to factor in how much that protein wants to break apart to. Actually, I got the algebra here if you really want. This actually, I said this in the textbook if you want to read it. I'm not going through this, but this rearranges to this by various steps. And what this is really representing is the idea that, like I say, that the protein is going to get more full if you supply it with more ligand. It's going to get less full if you supply it with less ligand. But you've also got to factor in the affinity that the protein has for its ligand. Because although if you supply it with ligand, it's going to come together, but also the protein has a tendency to break apart as well, because it's in equilibrium. It's going backwards and forwards. And so you need to factor in the backwards as well. And so that's what this KD is. Now, in 2 slides' time, this is confusing. In 2 slides' time, there's something that simplifies it a little bit. But just to make sure that it's clear to you guys, what KD is measuring is measuring the extent of dissociation. It's measuring how much it wants to break apart. And if that number is big, then it wants to break apart a lot. And if that number is small, then it wants to break apart a little. So if it's, as it gets further from 1, so it's 1 times 10 to the negative 15, that's reflecting that it really doesn't want to break apart at all. Whereas if that KD value is instead quite large, well, comparatively large, such as 2 times 10 to the negative 5, that's a larger number, closer to 1, then that's saying that it's not staying together as well. And this kind of graph and table is not test material, but rather it's flavor material to help illustrate the point that as you get a number closer to 1, which is technically a bigger number, well, not technically it is, but it's just a weird way of thinking about it, that's a lower affinity. A number closer to 1, which thus bigger, is indicating a low affinity, whereas a KD value with a small number further from 1 is indicating a high affinity. For example, enzymes bind their substrate quite loosely, but antibodies bind to the antigen comparatively stronger. And biotin, which you may have heard of, biotin, avidin, and streptavidin. If you do any research in biology and biochemistry, streptavidin will come in. That is where biotin, the ligand, will bind to avidin or streptavidin, the protein, with an extremely high KD value, extremely low KD value that is so low, so far from 1, so low, that it's almost considered such a tight interaction that doesn't want to break apart, that it's almost considered kind of covalent-like because it's so strong. But KD value, the point is that KD value is a range. If it's a small number further from 1, it's indicating tight binding. If it's a big number closer to 1, that's indicating loose binding. And if I go back a slide, back to this, then if you think about it, if the KD value is a big number, then you're dividing by a big number, and so Y is going to be smaller. If KD is a small number, then that means that it's not going to divide by much, and so Y is going to be a big number. So if KD is small, that's saying a high affinity, and you don't require as much ligand to fill it up. Or if KD is high, then you're dividing by a bigger number, and so more ligand is going to be required to fill it up. I got a little picture that I think helps to draw it together. And I like to use this little picture here. And often this is what helps click it into place for people. If you imagine that the protein is a bucket and the ligand is some water that you're pouring into that bucket. And I'm sure it makes sense to you guys that if you want to fill up a bucket, you got to pour water into it. And the more water you pour into it, the more full it gets. So Y is indicating the fullness of your bucket, and L is indicating the water that you're pouring in that bucket. And if you increase L, then you increase Y. If you increase the water that you pour into your bucket, then you increase the fullness of that bucket. Now, that's on one side. Now, that's not what happens, but I'm sure you guys are comfortable with the idea. You put in more water, it's going to get more full. What you have to compensate for the fact is the bucket has got holes in it. And it's got holes in it because the protein doesn't just come together, it also breaks apart. It's transient. And it has a tendency to break apart. Maybe it wants to break apart a lot. Maybe it wants to break apart a little. And so what you have to factor in now is that you have to factor in the rate of water coming in and the rate of water coming out. So you have to factor in the amount of the protein coming together and the amount of the protein breaking apart. And if you supply it with more ligand, then it's going to be more together. But it's still going to be, you're still going to be subtracting the amount that it's breaking apart. Now, if these holes are really big, that's indicating a greater KD value. If those holes are really small, that's indicating a smaller KD value. If those holes are really big, that's saying that you're going to be trying to fill up your bucket, but it's going to be losing ligand. It's going to be breaking apart more because it has a high KD value and it wants to break apart. If those holes are smaller, that's saying that it's not breaking apart more, so it's going to be easier to fill up the bucket. That's all it is. So if you have a really high KD value, what are you going to have to do to the concentration of L? This is a question I asked the class. I'll give everyone an opportunity to, if people could put their hands up, and then I'll pick someone. I'll give you like a couple of seconds to think about it. Let's be for a bonus point again. So if you look at, if KD value is high, what are you going to have to do to L in order to increase Y, which is the protein occupancy? I'll give you guys a couple of seconds to think about it. We've got one person. Anyone else want to give an answer? We've got two. Anyone else want to give an answer? I'll ask the question one more time. If the KD value is high, what are we going to have to do to L to increase Y? If the KD value is high, what do we have to do to L to increase Y? It's just Charles Anthony. So would you like to give us your answer?
Yeah, you have to increase L, right?
Yeah, you have to increase L. Thank you. So if you it's the same thing with like if you've got a bathtub that's got lots of holes in it, you're going to have to pour in water at a faster rate in order to fill up that bathtub. And if you don't, the bathtub's going to empty. That's all there is to it. So the KD value, a higher KD value is going to indicate that you're going to need more L in order to fill up the protein, whereas a low KD value is indicating that you're not losing much ligand, it's not breaking apart very much, it has a high affinity, so you don't require much ligand in order to fill up the protein. Now this becomes important as a little preview. Why do we care about this? Because the KD value for hemoglobin can change depending on the chemical environment. The chemical environment of the muscles is acidic from lactic acid, and that acid protonates some amino acids and causes the shape of the protein to change. And when the shape of the protein changes, the KD value changes. And what happens is that hemoglobin, when the acid comes along, it changes shape, and the KD value instead increases. If protons cause the KD value of hemoglobin O2 complex to increase, what's going to happen to the complex?
Can you repeat that?
Yeah, so if the hemoglobin complex, a hemoglobin oxygen complex, if the KD value increases because the environment has caused the KD value to increase, what's going to happen to the hemoglobin oxygen complex?
Would it decrease?
Yeah, and Charles Anthony has his hand up as well. I see that. But yes, it would decrease. It would break apart into hemoglobin and oxygen, and that's what happens. And so in the next lecture after the exam on Thursday next week, just before spring break, we'll start with hemoglobin. And essentially, all of the hemoglobin case study is just talking about the fact that the chemical environment changes the KD value of hemoglobin. And it changes the KD value so that the KD value is high at the site of the muscles because of lactic acid. Lactic acid causes the shape of hemoglobin to change, increase the KD value so it breaks apart, and thus if it breaks apart, then oxygen is released. And at the site of the lungs, you don't have that lactic acid. And so the protein changes shape and goes back to its previous shape with a low KD value. Since it has a low KD value, it does not require as much oxygen to fill up because KD is low. And that's it. That's it in a nutshell. Like the rest of this whole case study is just dressing that up and giving you more detail about it. But that's it. If you get that, then that's it. That's the lecture. So I'm going to keep going for a little longer, and I'm going to keep going to actually. I'm probably going to keep going just up to somewhere around about these slides. Just maybe the first 2 or 3 slides of this worked example. Got a couple more definitions, but we've got maybe 2 more slides of definitions and this equation here. And then about 2 more slides on this, and then I'll introduce the hemoglobin and we'll call it a day. So this is the next slide. So really, this is just, again, putting into words what I've just spoken. It's just saying that the ratio occupied is equal to the concentration of the ligand divided by the concentration of the ligand plus the amount that the ligand, the complex breaks down. occupancy is equal to ligand concentration divided by ligand concentration plus break apart. And if you increase ligand concentration, you increase occupancy. But you just have to factor in that it's going to break apart too. You just got to factor in that you've got you've pulled out the plug in your bathtub. And so if you want your bathtub to stay full, you're going to have to keep pouring water in there. And if you want it to get more full, you're going to have to put in more water. If you put in less water, then it's going to get less full. That's it. If KD is larger, then L needs to be greater to fill up the protein. That's it. Okay. And we can, as with most of these equations, we can put them onto a graph. And so this is just saying that Y, occupancy, against L, which is the X axis, which is ligand concentration. And you can see that as we increase the ligand concentration, we also increase the occupancy. And at first, it is quite rapid, but then after a while, it becomes saturated and slows down. And that's just due to the nature of a number divided by that number plus something else. If it's just kind of like Y equals A divided by A plus B, any equation like this is going to give a parabola curve like this. But that's just the math. You don't really need to know. The general thing, of course, is that you increase L, you increase Y. That's it. Now, an interesting fact of this, and this isn't a definition. People mix this up and say it's a definition. It's not a definition. The definition of KD is the definition would be the dissociation constant, which is the extent at which the protein will dissociate. It's the tendency of the protein. It describes the tendency of the protein to dissociate. It quantifies the tendency of the protein to dissociate. Now, that's the definition, but to calculate it, we can calculate KD, and KD is the concentration of ligand required to half fill the protein. So if we look at the concentration of the protein, if we look at the protein, we look at when it's half occupied, and we look at the concentration to half occupy it, that will be the KD value. That's not the definition of the KD value, but it's how we can measure the KD value. Sometimes in an exam, I'll say, what is the KD? Define the KD value. And someone will say the concentration at half occupancy. That's how you calculate it, but that's not the definition. The definition is the dissociation constant. It's the extent to which it breaks apart, but we can measure it in this way. And it's kind of an interesting quirk that you can do that, but it actually kind of makes sense as to why that is. In the next slide, I'll say why that is, and I believe that's the last slide of definitions. We got 15 minutes left of the lecture. I'm definitely not going to take up all of that time, but I'll just take up a little bit more. I feel like I say that every time, and I still do, but let's see. So again, calculate KD value as the you can calculate it from the amount of ligands required to half fill the protein. That's just how you calculate it. That's not its definition. The definition is a quantification, a measurement of how much the protein likes to break apart. The reason why it is the concentration required to half fill the protein is here. And it's because we think about it, if we set Y to being 0.5, then we've got 0.5 equals L equals L over L plus KD. And let's just change L to being X for now, because that just makes it easier to visualize. 0.5 equals X divided by X plus KD. And let's this time change KD into Y. Now, in order to balance this equation, what has Y got to equal? We can balance this equation, but what has Y got to equal? If I put it another way, 0.5 equals 1 divided by 1 plus x. Or let's stick it with y still to so it doesn't get 0.5 equals 1 divided by 1 plus y. What does y equal?
Negative 0.5?
No.
0.5?
No.
2, right?
The sum of is 2.
1.
1, yes. 1 divided by 2 equals 0.5. So 1 plus 1 divided by 1 plus 1 equals 0.5. So x divided by x plus y equals 0.5. Well, y must equal x, because x divided by x plus x equals 0.5. X divided by x plus x equals 0.5, because it's just a number divided by double that number is going to be half that number. So if we set y to being half occupancy, then whatever L is at that particular occupancy, KD must be that same number, which I think is actually, yeah. L must equal KD because L and KD must be the same number in order for y to equal 0.5. So that's that. So that is the definitions. I'm next going to move on to just introducing heme and hemoglobin and the problem and how biology solves that problem. But I can take questions about anything which I've talked about so far. I can go back over anything, re-explain anything, or if anyone has any clarifying questions about anything that I've covered so far. And I will say, for the winners of the quiz on Tuesday, I got a whole bag of various candies for the winning team last time. So there's your incentive for next time we have a class trivia like that, which will be just before exam 2. There will be another one where you can potentially win this booty of candies. And I believe our winners in the trivia, it was the lady sitting at the front, and their name was
The killer T cells.
Killer T cells. That was it. The winners, the killer T cells. I got a bunch, but also I don't want everyone to miss out, so I got a big pack of dum-dums for the whole class for the exam. Keep your blood sugar up while you're answering questions. So everyone gets a dum-dum on Thursday, but also there's a whole bunch of stuff for the winning team as well. I should also add on some bonus points. Well, everyone who participates and signed in with the attendance also gets a 100% quiz. So you get that. But it looks like no one wants to ask any questions about this this stuff here. It's important to have a nice. It's not super difficult, but it is a little fiddly. It may take a minute to get your head around, but it is important to have a good, firm grasp of this stuff, so that later on, when we're talking about KD value of hemoglobin and it changing, you know what we're talking about. I'm going to go on and just do maybe I think maybe 2 or 3 slides where there's a really nice point, and then I will stop it there. So, worked example using oxygen as a ligand, binding to heme as the protein, and this is an old slide that I haven't fixed that uses the British spelling instead, to form the protein ligand complex of hemoglobin, oxygen. And it's important to define, first of all, the way that things work is that you start out with oxygen, and oxygen is going to bind to iron. going to bind to an ion, which is going to bind to heme, which is going to bind to globin, which is our protein. And these are effectively cofactors. Cofactors are things that help proteins work, and the oxygen is our ligand. So the oxygen doesn't actually bind to the globin protein itself, but rather the oxygen binds to iron, which is held within a heme, which is held within a globin. So it's like a nest and a nest and a nest. It's like those little Russian dolls fitting inside each other. And the oxygen is bound to the iron in particular, but the iron is held by the heme, which is held by the globin. And we're going to talk a little bit about why that is. I'm actually going to largely skip over this slide here. This slide isn't it is kind of important. Let me think. I'm going to skip to this slide, and I'm going to define the problem. The problem is with oxygen is that oxygen is poorly soluble in aqueous solution, which is water, and that's because it's non-polar. Because although oxygen is polar, because it's two oxygens, it cancels out and it becomes non-polar. And because of that, non-polar things don't dissolve in water very well, and your blood is watery. So in your blood, only about 0.01, something like that, 0.04, I forget exactly, percent of your oxygen is dissolved in your blood. Because it's not, the oxygen isn't dissolved in your blood. Very, very little oxygen is dissolved in your blood. Instead, the oxygen is carried by a carrier. That's how you solve the problem. The problem which biology has is that it needs oxygen, but oxygen doesn't dissolve in the blood very well. How do we solve it? We have a protein carrier that carries the oxygen instead, because transition metals such as iron and an ion, 2+ ion, has a strong tendency to bind. Iron binds onto oxygen really, really well. Problem is that it binds onto it and it doesn't want to let go. And so how do we solve that problem? Well, the iron sits inside a heme, and you can see it here. The iron is sat within the heme in an organometallic complex, which is a porphyrin. And I'll go over this a little bit in more again next time. But it sits in the middle of the porphyrin ring, of the heme porphyrin ring, and it binds. Now this is that porphyrin ring, and we're looking at it instead of forwards, we're now looking at it sideways, right? Rather than looking at it face on like that, turning it sideways. So here it is sideways, and there's the iron sat in the middle of it, and there's the oxygen that's bound to it, perpendicular. And what happens is that this iron can move backwards and forwards. And when it moves forwards, it binds the oxygen more, but when it moves backwards, it binds the oxygen less. And when it moves forwards, Kd is decreased, and when it moves backwards, Kd is increased, reflecting that it is has a higher or lower affinity for oxygen. That is what's happening. And it kind of just looks like this, with the iron moving backwards and forwards in and out of plane of the porphyrin heme ring. It's being pulled by the protein. It's the protein which is pulling it and pushing it in and out of the heme ring, which is influencing its binding to oxygen. And that in turn is influenced by lactic acid in the muscles, signaling high metabolic activity, I need oxygen. That's essentially it. And we're going to talk about that in the next few lectures. I believe there's 2 lectures on this. We are up to slide 19, but I've kind of already explained this, but I'll go over it again. And we have just under 60 in total. So we'll do 20 slides in the next lecture, and then 20 slides in the lecture after that, or rather the next lecture. So that's going to be, of course, after the exam. So on Tuesday, we got the exam. Thursday, we'll do lecture 2 of hemoglobin and protein function. Then there's spring break, and then we come back, and there will be the final lecture on hemoglobin, and then we'll move on to enzymes. But that's everything for today. The attendance seems to be pretty good, so I'll just mark everyone as here, to be honest. But does anyone, you guys are free to go, but does anyone have any questions?
No, have a good day.
Awesome, great. Well, have a good day, everyone. Make sure you stay safe, stay warm, and
Have a good
we'll see you
day.
on Thursday for the exam. Oh, Zamir, you have a question?
Yes. Hold up. Okay, so earlier you said ligate versus substrate. What's the difference between a ligate and a substrate?
A ligand and a substrate. So a substrate is something that binds to an enzyme and will be changed to turn into a product, whereas a ligand binds and is released unchanged. So substrates bind, change into a product, and then are released. Ligands bind and are released unchanged.
Okay, and then I have one more question that kind of ties into that. So with the ligate doesn't the ligand doesn't change, so it'd be for the lock and key model, and then the substrate would be more used for like the induced fit.
No. When the changes that occur, for instance, with the induced fit are largely to do with things like bending, rather than like the chemical, elemental changes and chemical key chemical changes like swapping one group for another. It's not to do with that. Those are more to do with like the molecule just kind of bending. And so it hasn't, like, its shape has changed, but the molecule itself hasn't changed. It hasn't turned into a different molecule.
Okay, I get it.
But both enzymes and like other proteins that just bind, they both do a mixture of lock and key and induced fit. But induced fit more so, but they both do a mixture of the two.
Okay, thank you.
Great, no problem. All right, see you everyone. Have a good rest of your day. Yeah.
Oh, thank you again. Thank you.
No problem. I'm just figuring out how to end the Zoom call. I feel old now. I can't figure it out.